Okay, so today we're going to dive into what is basically a fascinating scientific detective story. It's all about how a team of researchers went on a mission to crack one family's cancer code and traced a deadly disease all the way back to a single typo in their DNA.
And it all kicks off with this really profound and, for so many people, deeply personal question. Why is it that some families seem to be haunted by cancer, generation after generation, while other families just aren't? This very mystery is what drives the entire investigation we're about to unpack.
And look, this isn't just some abstract idea. This right here, this is the real-life family tree at the very heart of our story. Every one of those red squares is a family member who is diagnosed with colorectal cancer. You can literally see the disease cutting a path across 3 generations. It's a devastating pattern that was just begging for an explanation.
Okay, so here's the thing. Scientists, they already know about some genes that can cause inherited cancer. But, and this is a huge but, for colorectal cancer, those known genes only explained about, 5 to 10% of the cases that run in families? That leaves a massive 90 to 95% of families, just like the one we just saw, completely in the dark, with no genetic answer. It's a huge puzzle.
So you got to appreciate the sheer scale of this challenge. We're talking about finding one single tiny error, a genetic typo, hidden somewhere in the 3 billion letters that make up a person's entire DNA. I mean, that is the ultimate needle in a haystack problem, right?
So how do you even start to find that needle? Well, these scientists basically became genetic detectives. Their main tool, something called whole genome sequencing. It's this super powerful technology that lets them read every single letter of a person's DNA code. They sequenced several family members, which gave them this ocean of raw data, but now they had to somehow make sense of it all.
To get through this mountain of data, they came up with a really ingenious strategy. They built this computational pipeline that acted like a giant funnel. It's designed to filter out all the irrelevant genetic noise so that only the most promising clues actually fall through. Let's walk through exactly how they did that. Okay, so get this. Their search started with a staggering number, almost 108,000 rare genetic variants. That's the initial pool of suspects. I mean, it's just an impossibly huge list to go through one by one.
So their first move was smart. It was based on this really simple but powerful idea. Whatever the genetic culprit is, it has to be in the family members who have cancer, right? So just by focusing on the variants they all shared, boom, they slashed that number down to 4,550. A huge cut, but yeah, they were still far from done.
All right, for the next step, they zoomed in on the parts of the genome that really matter the most, the genes that actually give the instructions for building proteins. See, a typo in one of these regions is way more likely to cause a real problem. And this was a game changer. It narrowed the field down to just 22 variants.
And for the final cut, they brought out the big guns, powerful computational tools to score each variant, basically predicting how likely it was to actually mess up the protein it was supposed to build. After all that sifting, that massive list was whittled down to just 3 prime suspects.
Just think about that for a second. From over 100,000 possibilities, down to just 3. This is that classic moment in any detective story, right? The investigators know they're closing in. It was time to look at the final lineup and figure out who the real culprit was.
And here they are, the lineup. The 3 final suspects were variants in these genes: OGF02, ZNF408, and SRC. But for the researchers, one name just leaped off the page: SRC. And you might ask why. Well, it's because unlike the others, SRC was already a known player in this world. It's a proto-oncogene, a gene that, when it goes haywire, is a known driver of cancer.
So what in the world is a proto-oncogene? The absolute best way to think about it is like the accelerator in your car. Normally, it helps your cells grow and divide in a nice controlled way. But if it gets a mutation, it can turn into what's called an oncogene, and that is literally like the accelerator getting stuck to the floor. You get uncontrolled growth, and that's the very definition of cancer.
And as they dug deeper, it got even more interesting. The scientists saw that this specific mutation, which they called V177M, wasn't just in some random part of the SRC gene. No way. It was smack dab in the middle of a critical region called the SH2 domain. You can think of this spot as the protein's main receiver. It's what listens for signals that tell the protein when to turn on and when to turn off. A mutation right there, that's like someone tampering with the main power switch.
Okay, so at this point, the SRC gene is definitely the prime suspect. But, and this is important, the case is still pretty circumstantial.
It's all based on computer models and predictions. To really get a conviction, our genetic detectives had to move out of the virtual world and into the lab. They had to prove in actual living cells that this gene was guilty.
And wow, this graph right here, this is their first piece of hard evidence, and it's pretty stunning. What they did was they took colon cancer cells, and they put this mutant version of the SRC gene inside them. And just look at that blue line. Those cells started growing way, way faster than the cells with the normal gene, which you can see there in yellow. This was the smoking gun. The mutation was absolutely causing uncontrolled growth.
So what was actually happening inside these cells to make them grow so fast? Let's go right back to our car analogy because it's perfect here. The lab experiment showed two big things were going wrong at the same time. First, the activity of the SRC protein itself was cranked way up. That's the accelerator being jammed to the floor. And at the same time, the levels of a really important tumor suppressor protein called P53 went way down. Now, P53 is one of the cell's main emergency brakes. So think about it. The accelerator is stuck on and the brakes are failing. That is a recipe for disaster.
And here you can see the actual proof from the lab that confirms our analogy. These images, they're called Western blots, are basically a way for scientists to measure how much of a specific protein is in a cell. See that darker band up top? That tells you that the activated form of SRC is much higher in the cells with the mutation. And then, see the lighter band at the bottom? That shows the protective P53 protein is much lower. It's the visual confirmation of that whole accelerator and brakes problem we were just talking about.
So, with all the evidence on the table, it was time for the verdict. The computer data pointed to a suspect, and the lab experiments proved it was guilty, really, beyond a reasonable doubt. But what does this all mean? Not just for this one family, but for how we understand cancer as a whole.
This diagram here really explains that final crucial piece of the puzzle, how it all works. See, the normal SRC protein has this amazing built-in safety feature. It's like an internal off switch, where one part of the protein folds over and latches onto another part to keep it turned off. But this single mutation, this V177M, it completely disrupts that connection. It basically breaks the off switch, forcing the protein to get stuck in a permanently on state, just constantly screaming at the cell to grow, grow, grow.
And so, after all that incredible detective work, we get to the official conclusion from the scientific paper itself. They state that this specific variant, SRC V177M, is now identified as a new genetic factor that predisposes families to colorectal cancer. A huge breakthrough.
So let's zoom out. What's the big picture takeaway from all of this?
Well, first, it does more than just solve a mystery for one family. It adds a critical new piece to this giant puzzle of hereditary cancer. It also helps us understand this idea of moderate risk genes, which helps explain why not everyone with the mutation gets cancer. And maybe most importantly, by showing us exactly how this broken off switch works, it opens up a whole new door for researchers to potentially develop new ways to prevent and even treat this awful disease.
You know, this whole journey, starting with a family's painful history and tracing it all the way down to a single letter in their DNA, it just leaves you with this final, really powerful question. It really makes you wonder, for the thousands of other families out there who are still searching for answers, how many other genetic mysteries like this one are just waiting to be solved?